Hi, everyone. Welcome to my homeschool compliments of the coronavirus. I hope you and your families are all doing well. I'm sure at this point you're getting bombarded by many emails from your professors, including me. So I hope you do have some time to go through all those and read those. And I'm not going to take the time to go through all the announcements here, but make sure you read those so that you know how this new format is going to work to finish out this semester. And whatever you do, do not worry. We'll get through this one way or another, and it will be fine. I will do my very, very best to deliver the best instruction I can with this new format. So keep in touch with me and it'll all be good. So today we are going to pick up where we left off, and we were talking last time I saw you face to face about mitosis and meiosis and how cells divide, and specifically in meiosis, how cells divide in order to make gametes, that is, egg cells and sperm cells. Today we're going to look at how those gametes come together to make individuals and what's the chance of passing on different traits to our offspring. And we're going to begin with a look at what we call Mendelian genetics. Gregor Mendel was a monk who did the first work in this field, so we'll be talking about him and some of his simple genetic studies, and then we'll look at some exceptions to those rules later. But chapter 14 is essentially looking at basic Mendelian genetics. So this is page 79 in your course outline and page 269 in the text. And starting with concept 13.1, this is actually from chapter 13, but I wanted to mention it because it does seem relative to moving in a good transition into moving into chapter 14. And it starts there talking about how offspring acquire genes from parents by inheriting chromosomes. And in a literal sense, children do not inherit particular physical traits from their parents. It's the genes that are actually inherited. And so what are genes? Genes are actually pieces or segments of DNA that code for proteins. So when we're talking about these traits, we're actually talking about proteins. So if you talk, for instance, about skin color, eye color, or hair color, that is the protein melanin. If you're talking about the trait blood type, for instance, that's the protein on the red blood cell. So again, genes are the pieces of DNA that code for these proteins. And they are passed to the next generation via reproductive cells called gametes, which we're already familiar with, sperm cells and egg cells. And I saw this article in the National Geographic, the special issue talking about these twin sisters that make us rethink everything we know about race. And I thought, with all the social problems caused by skin color, from a biological perspective, it's not so black and white. So I wanted to share this video with you because, as you can see here, these two girls, if you saw them on the street, you might think one's black and one's white, but actually they are very genetically similar. And yet, their skin color looks this different. So it's a lot more complicated, and as we get into these different traits, like skin color, that are not just black and white, we'll see how confusing and complicated it can be to try to make these predictions. So here's a clip on this.
Hosting a dinner party for your friends can be tough. So, here's the thing. No one's gonna know it's store-bought.
My name is Marcia and I'm 11 years old.
My name is Minnie and I'm 11 years old.
When they see us together, they don't believe that we're twins.
They don't believe us, like they're like, oh really, I never noticed that, like I thought you just friends.
Because we're a different colour.
Racism is where somebody judges you by your colour and not like by your actual self.
I think racism is a negative thing because sometimes it can hurt people's feelings.
Just because you're a different colour doesn't mean that you have to change the way you are just to be like someone else.
If the world has to say anything about us, then if it's going to be a rude comment, then don't bother say it.
I would say just don't listen to them. Just ignore them, pretend they're not there, but they disappeared.
If it's going to be a nice comment, then go ahead.
I will tell them to not, like the people say, don't judge a book by its cover, don't judge other people by their looks, because there could be something totally different on the inside.
If you're happy with the way you look, then you should stay the way you look.
Just be yourself.
I love that. And it's so true because again, this skin color is just a matter of combining different genes that were carried through gametes. So again, we'll get to the exceptions to regular Mendelian genetics in the next chapter. But for now, looking at concept 14.1, where Mendel used a scientific approach to identify two laws of inheritance. And he discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. And he was very lucky that he chose garden peas because they are very advantageous, mostly because for every trait he studied, there were only two different forms. Unlike us, when we look at eye color, for instance, or even skin color, there's many different shades. And so he started out doing these single trait experiments. I'm going to show you this clip, and then we will talk about how these are done.
This is Tyler. Tyler, with Grammarly's help, is writing an email to his boss, Anita.
Mendel used self-pollinating pea plants in his experiments because their flowers, unlike those of many other flowering plants, are created in such a way that flower petals completely cover each flower's sexual structures so pollen can't be distributed between flowers by wind or insects. But pea plants can be cross-fertilized by hand. Mendel would carefully open the flower of the plant to be fertilized and cut off its male sexual structures so that the flower couldn't self-fertilize. Then he would carefully collect pollen from a flower belonging to a second plant with which he wished to breed the first plant, and then apply this pollen to the female reproductive structures of the first flower. Mendel hypothesized that as he transferred pollen from the male reproductive structures of pea plants with white flowers to the female reproductive structures of pea plants with purple flowers, he was bringing together discrete physical units, one from each parent, which formed pairs that together would determine flower color in the next generation of pea plants. Mendel referred to these discrete physical units as factors. Today, we call them genes, and know, unlike Mendel, that they are made up of segments of DNA found on even larger pieces of DNA called chromosomes. The different forms that a gene can take are referred to as alleles. In Mendel's flower experiment, there were two alleles, one for purple flowers and one for white.
Okay, so one of the first thing you'll notice that when Mendel worked with these garden peas, he always started with what we call pure parents of contrasting characters. And he called those the P1 generation. The P stands for parent of the first generation. And when he talked about contrasting characters, for instance, as I mentioned, with pea plants, for every trait he studied, like maybe seed texture, he found there were only two forms. In this example, it would be these pea plant seeds either were smooth or wrinkled, just two different forms. And so he called those pure parents of contrasting characters. And when he bred them, the offspring produced, he called the F1 generation. That F stands for filial or family. And then he would grow those and cross those first generation plants and have them self fertilize. So it's a form of inbreeding. And then he would get the F2 generation. So, in other words, the F1 seeds became the P2s, the parents of the second generation. So, the children became the parents. And then those parents' kids were called the F2s. So, and he could go on and on, but essentially, uh, we're just going to look at the F1s and the F2 generations here. So again, looking at how he used these pea plants, these were unique also because the petals actually made it very difficult for them to cross fertilize. And by that I mean if you look at a perfect flower, we have both male and female parts within the same flower. This part right here is the female part in the center. This is called the stigma style, and then the ovary with ovules down here. And then surrounding it, all these little structures are the anthers and filaments. And the anther has pollen and pollen has sperm. So these, if left alone, if you had a group of these purple flowers, they would self-fertilize and you would always get purple color showing up if you were studying the trait of flower color. So he did that until he was sure they were pure breeding for purple. And then he did the same thing with the white. And so he had what he called pure appearance of contrasting characters, all purple flowers and all white flowers. And that, again, these were the parent generation, and that would make up the F1, the seeds for the first generation. And he noticed when he planted those seeds, the first generation all showed purple flowers. So he started wondering, since one parent was white and one parent was purple, why did all the F1s come up purple? What happened to the white? The white seemed to disappear. And so he came up with the principle of dominance. The principle of dominance is basically saying that one of those traits is dominant over the other. In this case, the purple was dominant over the white. So the one that showed up, he called dominant, and the one that was hidden, he called recessive. By the way, there's lots of terms in this section, so make sure that you are making flashcards for all of these bolded words in your course outline. So, looking at this again, he started with pure appearance of contrasting characters, purple versus white. He called them true breeding. We now call them homozygous. Homo meaning same. They all contain the same trait for purple in this batch and all white in this batch. And then in the first generation, they were all purple. That was a principle of dominance, but he figured that white was in there somewhere. So how about if the F1s become the P2s? We're going to take some of these purples, which he called hybrids, because they were a result of parents of contrasting characters, which now we call heterozygotes, meaning they're going to have a gene for each form of these traits, and he crossed those, and he got the F2. So these purple ones became the F2s, and their offspring became, the purple ones became the P2s, and their offspring became the F2 generation. And when he counted them up, he noticed there was a trend for ratios, and he always got roughly a 3-to-1 ratio for purple versus white. But look, the white one did show up. So he figured then that that white must have segregated out in the second generation. And that led to the principle of segregation, also known as the first law of heredity. When Mendel crossed the F1 hybrids, many of those F2s had purple flowers, but some had white. So again, this principle of segregation is saying the white that was hidden in the first generation can segregate out and show up in about a quarter of the F2s. So he discovered this ratio of about 3-to-1 in the F2 generation, purple to white. So he did this with all kinds of traits. And again, that's why he was lucky when he picked the garden plant or the garden pea, because they always had only two different forms. For flower color, purple was dominant over white. For flower position, axial, which was dominant axial down here versus terminal at the tip, was recessive. Yellow was dominant over green, and round was dominant over wrinkled when it came to the seeds. And for pod shaped, inflated was dominant over constricted, and so forth. And notice these ratios, they're not exact, they're about a 3-to-1 ratio every time. So in the genetics lab, that's one of the things we explore is when you count these, you're never going to have exactly 100 plants with 75 showing the dominant and 25 showing the recessive. So when you get these kinds of numbers, you have to run what is called a chi-square test. It's a statistical analysis to decide, is 428 to 152 actually a 3-to-1 ratio? Is it close enough for us to say yes? So we'll get to that later. So more terms here are, this new term, allele, is an alternative version of a gene. So again, every trait that Mendel studied, he noticed there were at least two different forms. In the garden pea, it was always two different forms. Purple in this case versus white. So the one form, purple, is one allele, and the other form, white, is another allele. So this on a pair of homologous chromosomes, remember, homologous chromosomes, homo again means same, these are coding for the same trait, but they might be different alleles or different forms of a trait. So we have terms to describe it when they're the same and when they're different. And again, that word homo means same, hetero means different. Another term here that we need to define is what is a gene locus, or loci is plural for that. So when we look at the location for the gene for flower colors down here and seed colors over here, that's simply the gene locus. That stands for location. So more terms here. When the two alleles are the same, that is, they're both purple or they're both white, from each parent donated the same form, each one donated an allele for purple or an allele for white, we call that homozygous, homo meaning the same. When they're different, we call that heterozygous. And we always know if it's heterozygous, you get one of each. So these letters represent the alleles. And the letter itself represents the gene, and capital and lowercase represent the different forms. So if we say gene A is flower color, and the capital letter would be purple, which we know is dominant over the lowercase letter, which is white, then if the plant was homozygous, it would either be big A, or little a. The same, though. It has to be the same letters. Same as in capital or lowercase. If it's heterozygous, you know it's got to have one of each. So these letters represent what we call genotypes. Genotypes are the actual genes present. Homozygous dominant would be two capital letters. Homozygous recessive, two lowercase letters, and one of each is called heterozygous. Not to be confused with the phenotype. The phenotype is the physical appearance. How does it actually look? Is it purple or is it white? So anytime you get questions asking for phenotypes versus genotypes, phenotypes, what does it look like? Purple or white? Genotype, you always have these three choices. Is it homozygous dominant, heterozygous, or homozygous recessive? One thing you should never say when asked about genotypes is the answer is AA, because guess what? They're all AAs. So you have to use these terms. Homozygous dominant means two capital A's. Homozygous recessive means two lowercase a's, and heterozygous is one of each. And this is also important when you do your Punnett squares to pick letters where you can tell the difference in a capital versus lowercase. So if you're typing, it's easy to tell with any letter, but if you're printing, it's hard to tell sometimes the difference between a capital P and a lowercase p, or an S, or an X. And so again, any of those letters that are too similar, you should not use. So I always just stick with A's or B's. So when we looked at the phenotype of his F1, where he got the 3-to-1 ratio, sorry, F2s, it came out a 3-to-1 ratio of purple to white. Those are the phenotypes. But when we look at the actual genotypes, notice it's a different ratio. It's a 1 to 2 to 1 ratio. We got 1/4 that are homozygous dominant. And again, if you're typing, you can use P's. Your author uses P's. I prefer A's because capital P's and lowercase are hard to tell the difference. But anyway, homozygous dominant, you got 1 out of 4 chances. Heterozygous, you have 2 out of 4 chances, and homozygous recessive, 1 out of 4. So what we want to do then is show how Mendel got these ratios by doing Punnett squares. How did he get the 3-to-1 phenotypic ratio and the 1 to 2 to 1 genotypic ratio? So the possible combinations of sperm and egg can be shown in a Punnett square. So what is a Punnett square? A Punnett square is actually a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. Couple things to remember here. It's a prediction. It's not saying that you're going to have 4 kids. One's going to be this, one's going to be that. It's saying that you have a 1 in 4 chance with every kid of having a certain trait showing up. Also, we are only looking at one trait right now. And so when we do these one trait crosses, known as monohybrid crosses, mono means one, we want to use a capital letter for the dominant allele, lowercase letter for the recessive, and keep in mind when you do these Punnett squares, you're going to have 2 parents, each can have a capital or a lowercase, and since you have 2 parents and each can have 2 options, a dominant or recessive allele, you're going to have a 2x2 box or a box of 4. So this is for only monohybrid crosses. You use a 2x2 box, you put the parents' alleles across the top and down the side, and then you bring the ones at the top down, the ones at the side across, and that fills in the boxes. These boxes are for the F1s. These are the parents, these are the children. Again, this doesn't mean you're going to have 4 kids. It means that you have a 1 in 4 chance of having this genotype, homozygous dominant, 2 out of 4 chances of having a heterozygous condition, and 1 out of 4 of having a homozygous recessive condition. Now, another thing when looking at these boxes is as long as there's a capital R, and let's say that R stands for red and the little r stands for white, we would say that these flowers, as long as there's a capital R, these would all be red. The only time you get the recessive color showing up, that is white, is if they're both the same lowercase letters. As long as there's one R, remember the principle of dominance that tells us the capital letter is dominant over the recessive. So phenotypically, it would be a 3-to-1 ratio, 3 red and 1 white. But genotypically, it would be a 1 to 2 to 1 ratio, 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So it's kind of like flipping a coin. Keep in mind that the coin has 2 sides. It could be a dominant or recessive allele if you change the heads and tails to big R, little r, and you have 2 parents. So since you have 2 parents, the male and the female, each one could have a dominant or recessive. Again, you need a 2x2 box. So always with monohybrid crosses, we are going to set up this 2x2 box. So if you follow these rules, you will always do this correctly. And you will notice in your course outline, I have some practice problems on page 87 and 88, and the answers are on page 89 and 90, as well as if you want to see the actual Punnett squares, if you go to Canvas under handouts, you'll see the actual Punnett squares. But you should definitely practice doing all the practice problems that I've given you before you take the first test. And I'll tell you more about the first test later. But again, if you follow these directions, you'll always get it correct. So you want to choose a symbol, a letter, for each trait, and use a capital letter for dominant and lowercase letter for recessive and show them in a key. So let's say we're going to say big R equals red, little r equals white, and then show the actual cross. Well, in this case, they're using letters A, but we could change these to capital R's and lowercase r's. The point being that we're going to start with pure appearance of contrasting characters, which means homozygous dominant and homozygous recessive. Draw a Punnett square. That's the 2 by 2 box. So you go 1, 2, and then 1, 2 to get this 2 by 2 box. And put one parent's alleles across the top, one parent's down the side. Fill in the boxes by bringing those at the top down and those at the side across. And find the phenotypic and genotypic ratios. Again, if it's a phenotype, if the question asks about phenotype, you want to definitely go back to the key and ask yourself, is it purple or white? Or in this case, is it red or white? If it's a genotype, you have three choices. Is it homozygous dominant, heterozygous, or homozygous recessive? So here's our first question. Cross a purple and white pea plant. And we're going to set it up just like this with pure parents of contrasting characters. We're going to show Mendel's principle of dominance and principle of segregation using these Punnett squares. So now I am going to go to the whiteboard and hope all is well on my whiteboard and that nothing falls down. Okay, so as it says, choose a symbol, a letter for each trait, and using a capital letter for dominant and a lowercase letter for recessive, show them in a key. So I'm going to use A's, and I'm going to say big A, and we're crossing a purple and white. So big A is purple, and little a is white. Okay, and then it says show the actual cross of the P1. So the P1, here's my cross. We're going to cross pure appearance of contrasting characters because we're trying to prove Mendel's principle of dominance and segregation. Then it says draw a Punnett square and put one parent's alleles across the top and the others down the side. So when we draw a Punnett square, 2 by 2 box, so it's going to go 1, 2, 1, 2. One parent's alleles across the top. It doesn't matter which one goes where. Just pick one and put it one across the top and the other one down the side. Bring those at the top down and those at the side across. Okay, and this is going to be our F1. Now, do you always have to put the capital letter first? I recommend that because remember the principle of dominance. As long as there's one big A, it's going to show up as that, in this case purple, over the white. So now the question is, what are the genotypic and phenotypic ratios? You can do ratios or you can do percentages. Since these are all the same, I like to use percentages because that's not really a ratio. So I'm going to say for my phenotypic, remember phenotype, go back to the key, is it purple or white? And they all have a capital A, so they're going to be 100% purple. Now for the genotype, you have three choices. Are they homozygous dominant, heterozygous, or homozygous recessive? Since they're all the same, these are 100% heterozygous. Okay, and there is our F1 results. But remember, the F1s became the P2s. So this is the principle of dominance that says all of the F1s show only one trait. But now moving on to the F2s, remember the F1s become the P2s. So now we're going to take any of these, it doesn't matter, they're all the same. And I'm going to do a 2 by 2 box. I'm going to use the same key, purple and white here. Bring these down and these across. And notice this would be now the F2s. So What about our phenotypes? Again, if for phenotypes, go back to the key. What percentage are purple? What percentage are white? You could do a ratio, but again, I'm going to stick with percentages. And I'm going to say as long as they have a big A, remember they're purple, so they're going to be a 3-to-1 or 75% purple and 25% white. Okay? And if I look at my genotype, it's going to be a little bit different. Because for the genotype, you've got now what percentage are homozygous dominant? That would be 2 big A's. So we've got 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive. So this is our 3-to-1 ratio, which is the same as our principle, or which proves our principle of segregation, which says the recessive allele, which was hidden in the first generation, can segregate out and show up in 75% or 3 out of 4, or sorry, 25% of the population. Principle of segregation referring specifically to the homozygous recessive. Now remember, these principles, the principle of dominance and the principle of segregation, are only based on the phenotype, because Mendel only knew what he saw. He didn't know about actual alleles or genotypes back then. So that's those principles, and we have just proved them. So once again, just looking at these two principles, the principle of dominance and the principle of segregation. And you can see here again what we did. We started with pure appearance of contrasting characters, and in the first generation, they were all heterozygous. Again, phenotypically, the appearance was 100% purple. That's a principle of dominance. And then in the F2, we got that 3-to-1 ratio, 25% of the population showing up with that recessive trait. So that's the principle of segregation, saying that the recessive allele, which was hidden in the first generation, segregated out and showed up in 25% of the population, or a 3-to-1 ratio. Okay, so the next question is, how can we tell the genotype of an individual with a dominant phenotype? And this is something we actually did in lab. We grew some corn seedlings where green is dominant over albino. You can see some of these white ones here are albino. And the green ones we know are dominant. They have at least one dominant allele, but we don't know if they're homozygous or heterozygous. And so what you could do in a case like this is you could carry out a test cross where you could take one of these green ones and cross it with a homozygous recessive white. We know the albino has to be homozygous recessive, because if it had one dominant allele, it would be green. So the only way homozygous recessive conditions show up is in a case where they have only the recessive alleles. So if you cross that green with a white and you get even one white showing up, then you know that green seedling also had to have a recessive allele, which means it had to have a dominant allele to be green. It had to have a recessive allele for it to have a recessive offspring showing up, which means it has to be heterozygous. So here's how it works. So if you're trying to figure out whether this purple plant is homozygous dominant or heterozygous, what you would do is cross it with the white one. And notice if they always show up purple, most likely it's homozygous dominant. But we don't know that for sure, because even if it's heterozygous, it still has a 50-50 chance of having a purple. But if we cross it with a purple and get even one white flower showing up, we know that parent had to have at least one recessive allele. So you always know if a dominant parent has a recessive offspring, that dominant parent has to be heterozygous. It's weird for me not to be asking you questions as I go through this, so email me your questions. I'll also have live chats set up so that we can communicate that way as well, and I'll be sending you emails about that later. Okay, so that concludes the monohybrid crosses if we're just looking at one trait crosses. But Mendel got to thinking, well, if all the purple or when I looked at all those dominant traits, and then I looked at all those recessive traits, I wonder if they are inherited together. In other words, if the plant has one dominant trait, does it have to have all dominant traits? It's kind of like saying, do all blondes have to have blue eyes and all dark-haired people have to have brown eyes? So he started looking at what we call two trait crosses, and these are known as dihybrid crosses, di meaning two. And he set up the experiments the same way. He started with pure appearance of contrasting characters only for two traits. So this time he's looking at seed color and seed texture, where yellow is dominant over green and round is dominant over wrinkle. And he used the Y to represent yellow. Again, not a great letter if you're handwriting this stuff, because the capital and lowercase sometimes look the same. R is a pretty good letter, though. And he started with pure appearance and contrasting characters for both traits. In other words, they were homozygous dominant for both traits, yellow and round, and homozygous recessive for both traits, green and wrinkled. And when he crossed them, guess what? The first generation showed the same as the monohybrid cross. They all had the dominant trait showing up. But just like he did with the monohybrid cross, he carried them out to the F2s, and this time he got a very interesting ratio down here, which we call the 9-3-3-1 ratio. 9/16 had the dominant trait, 1/16 the minority had the recessive traits, and the 3/16 were a combination of one dominant and one recessive. So how do we get that? And how do we do a Punnett square when it comes to dihybrid crosses? Now things get a little bit more complicated. And Mendel then identified his second law of inheritance by following two characters. Again, this is a dihybrid cross. And he came up with, in that second generation, when he got the 9-3-3-1 ratio, he called this the law of independent assortment. And this is our second law of heredity that says that these genes can assort independently. That is, they're inherited independently. Just because you inherit a dominant for flower color doesn't mean you have to be dominant for seed texture or whatever it is. So it gets a little more confusing now because we have two traits, so we're going to have to add an extra step when we do these monohybrid or dihybrid crosses. So we're going to start with crossing a homozygous purple flower with a flower tall plant where A is tall, capital A is tall, little a is dwarf, and then a tall plant where B is going to be, capital B is the tall plant, little b is the dwarf plant. And so back to the whiteboard for this. I'm hoping my whiteboard does not fall down because I didn't put it up permanently. I only will be using it for these genetic problems. Okay, so we have to do the same thing that we did with the monohybrid cross in terms of following these steps. We're going to start with the traits and show them in a key using a capital letter. So we're going to say capital letter is tall, capital A, lowercase a is dwarf. And for the other trait we were looking at is, oops, sorry, did that wrong. We were going to do capital A is, flower color. So this is purple and white. And then they use B is going to be tall versus dwarf. Okay. Now, in this case, you have an extra step. And so we're starting our parent cross for two traits, pure parents of contrasting characters for two traits. That means homozygous for both capital A's and B's, and homozygous recessive for the other parent. And the step that you have to add here is what I call FOIL. Most of you probably remember this from algebra, first, outside, inside, last. And it's saying, because of our principle of independent assortment, that these A's and these B's do not have to be inherited together. They can assort out independently. So we have to show what are the possible allelic combinations in the gametes. In other words, when these separate out into separate gametes in meiosis, what is the possible combination? And so you separate the A's from the B's and foiling, and you're going to put the first A with the first B, the outside A with the outside B, the inside A with the inside B, and the last A with the last B. And notice these are all the same, so we can cross out any repeats because this parent only has one option in its gametes, big A, little b, or big A, big B. And we do the same for the other parent. First A with first B, outside A with outside B, inside A with inside B, and last A with last B. Cross out the repeats, and guess what? I only need a 1 by 1 box. I only have one possible allelic combination for this parent and one for this parent. Now, if you didn't cross out your repeats, you would have a 4 by 4 box, which is a lot more work, but you would still get the same results. So I just crossed those out because we really only need a 1 by 1 box. And 1 by 1, showing one parent's alleles across the top, one parent's on the side. Now, a couple things to notice in a dihybrid cross. Di meaning two. Each parent box has two alleles, and the offspring are going to have four. You have to keep your A's together and your B's together. And you can see here, because they both have a capital letter, and at this point, for all the rest of these, we're only going to stick with phenotypes. The only time I'm going to ask you about genotypes is in a monohybrid cross. So what are the phenotypes? Remember, the phenotypes, you go back to the key. Since there's a big A, it's going to be purple. Since there's a big B, it's going to be tall. So these would be 100% purple, tall. All right, this is the F1. They're all the same. Now the F1 becomes what? The P2s. P2s. So they're all the same, so we're going to cross two of these parents in this next section. So now when we foil, we've got first A with first B, outside A with outside B, inside A with inside B, last A with last B. Do I have any repeats? No. Uh-oh. Now I need 4 boxes for this parent. And likewise, this is the exact same thing, so I'm going to have the same thing. So I need 4 boxes for this parent, which means now I need a 4 by 4 box. I hope I can fit this. I'm going to try. So 1, 2, 3, 4. 1, 2, 3, 4. Getting close. One parent. Now remember, dihybrid means make sure you put 2 letters in the parent boxes. And then this one down the side. Now I'm not going to fill this entire thing out for you because I want you to practice this. I'm going to fill out a few of them though. Bring these down and across. Keep your A's together and B's together. This one's going to be little A's and big B's. And this last box would be all lowercase letters. But here's the thing. Since I already know anytime I have a 4x4 box, this is going to be Mendel's principle or law of independent assortment, what ratio am I going to get? I'm always going to get I'll write it down here, 9:3:3:1 ratio. That's the principle of independent assortment, which tells us that 9/16 of our offspring are going to, in other words, the majority here are going to have both the dominant traits, purple tall. 1/16, the minority, will have the recessive traits, white and dwarf, and then 3/16 will be a combination of either purple and dwarf or white and tall. So keep that in mind that you don't have to write out that whole or finish that whole box. Once you see it's a 4x4 box, you know right away you're going to get that 9:3:3:1 ratio. Okay, hope everyone is still with me. And at this point, we'll take a break, let you finish that box out, and then we will continue on in the next video with the rest of these genetic problems. And I will see you then.